We're delighted to announce that Anne-Laure Fameau, a researcher at L'Oréal France, will be the recipient of the 2021 AOCS Young Scientist Research Award, sponsored by the International Food Science Center. For more than 20 years, the AOCS Young Scientist Research Award has recognized scientists who have already made significant and substantial research contributions in areas relevant to AOCS. Dr. Fameau received her PhD in physical chemistry from the University of Nantes, France in 2011. She worked at the French National Institute of Agricultural and Environment Research in Nantes and carried out research at North Carolina State University in the United States and the Technische Universität Berlin in Germany before joining the physical chemistry department at L'Oréal in 2015. Dr. Fameau's research focuses on responsive soft materials made from lipids and green surfactants, with a particular emphasis on foams and interfaces. After synthesizing these materials, she develops detailed structural characterizations of them using X-ray and neutron scattering techniques. She has identified the presence of capillary forces at the nanoscale that reveal a fundamental assembly mechanism and highlight potential applications for soft materials in consumer products. She has published 38 peer-reviewed papers and reviews and 3 book chapters. She received the European Young Lipid Scientist Award in 2018 and was a plenary speaker at the International Conference on Small Angle Scattering in Berlin, Germany in 2015. Dr. Fameau is recognized for her fundamental and applied contributions to the emerging area of oil foams and for her outstanding contributions to the physical chemistry of lipids in cosmetic science and foods. We also recognize her sustained contributions to outreach and her service to our community. Congratulations, Dr. Fameau.
Good morning. My name is Alex Marangoni. I'm a professor and Canada Research Chair in Food Health and Aging at the University of Guelph in Guelph, Ontario, Canada. And today I have the great pleasure to introduce to you our award winner for the Young Scientist Research Award of the American Alchemist Society, Dr. Anne-Laure Fameau. She is a researcher working at L'Oréal in Paris, France. Her research interests are in responsive soft materials based on lipids and green surfactants, particularly fatty acids, with an emphasis on foams and interfaces. She has done some recent, very interesting work on oleofoams lately as well. She couples the development of these systems with detailed structural characterization using X-ray diffraction, neutron scattering techniques, and a lot of this work is taking place at synchrotrons. Her work has demonstrated the presence of capillary forces at the nanoscale, revealing a fundamental assembly mechanism and highlighted new potential for sustainable materials in consumer products. She has published 38 peer-reviewed papers and reviews and 3 book chapters. Among those, of course, a paper in Nature Materials, which you should be very proud of. She recently received the European Young Scientist Award in 2018, and was a plenary speaker at the International Conference on Small Angle Scattering in Berlin, Germany in 2015. Dr. Fameau enjoys transmitting her passion for science to young researchers as well. She has visited many schools and given scientific talks to students to promote science in Paris and especially to promote science for girls. Thank you very much for joining us. And with that, I will turn it over to Dr. Fameau.
Thanks a lot, Alejandro, for this very, very nice introduction. And first of all, I would like to thank the AOCS committee for giving me this award. I'm very, very honored to receive this award. I'm very, very happy. It's really a great honor. And I would like also to thank the people who nominated me and who supported my nomination. Thanks to all of you. And today I want to show you how we can use self and directed assembly to design smart materials based on fatty acids. I really hope that after my presentation, you will be convinced that fatty acid soaps, even if they are known and used in centuries, they have still a plethora of physical chemical properties that need to be studied and used to design new materials, such as, for example, smart materials. And it's really fatty acid soap is still an open area for research. So the results that I'm going to show you today have been obtained in different parts of the world. So most of the results have been obtained in France when I was working in my institute in the French National Institute of Agricultural and Environmental Research in RAE, in collaboration with the French Neutron Center, the Laboratoire Léon Briouin in Paris. Some of the results have been obtained when I was working in the group of Professor Maria Cristina Agnon in Argentina in the University La Plata. All the results that I'm going to show you today about magnetic field assembly have been obtained when I was a visiting scientist in a group of Professor Orlin Velef at NC State University in the US. And some of the results that I'm going to show you about photoresponsive foams have been obtained when I was working in the group of Professor Regine von Klinzing at the Technische Universität Berlin. So the main question is how to develop multi-responsive soft materials based on fatty acids. The answer is quite simple. You need to use colloidal assembly. But with colloidal assembly, you have two different strategies. You have the self-assembly strategy, or you have the directed assembly strategy. And today I'm going to show you one example of how you can use self-assembly based on fatty acid soaps to design multi-responsive aqueous foams. And I'm going to show you an example of how you can use directed assembly based on fatty acid soaps to develop ultra-flexible magnetic filaments. So first, we are going to begin with the self-assembly approach and the design of responsive aqueous foams. So why it is important to study foams? Because foams are present everywhere in our everyday life. As for example, as food products, such as shown here, chocolate mousse or foam beer, or as cosmetic products, such as shampoo, shaving foam. Foams are also very important as foam extinguisher or to decontaminate surfaces. And I really like sport, and especially soccer and football, and foams are also very useful in soccer because, as you can see here on this picture, the referee is using a foam to draw a white line to be sure that this guy here will keep their distance with the goalkeepers. So as you can see, foams are present everywhere in our everyday lives. But what is a foam? So a foam is a dispersion of a gas bubble into a liquid phase. And this liquid phase forms the so-called plateau borders and the thin film that separate the bubble. And this liquid phase contains the foam stabilizers, that is to say surfactants, particles, polymers, or protein that absorb at the gas-liquid interface and stabilize the bubbles. So if you want to understand how a foam is stabilized, you need to study the foam at the different length scales, at the microscopic scale of the foam, at the plateau border scale, thin film scale, and at the interface scale. The main problem with the foam, it's like a foam is an unstable system that tends to destabilize with time under three main mechanisms of foam destabilization. So the first one is called drainage. It's due to the gravity. Due to the gravity, the liquid flows through the foam from the top to the bottom, and the result is a dry foam. The second one is called coarsening. So when you have two bubbles, when the smaller bubbles just transfer the gas to the bigger one due to the difference of Laplace pressure between the smaller bubbles and the bigger bubbles. And the result is a disappearance of the smaller bubbles. And so you have an increase of the bubble size with time. One is called coalescence. So when you have two bubbles, when they enter in close contact, the thin film between the bubbles can just break, and the two bubbles just reorganize to form one single bubble. So again, the result is an increase of the bubble size with time. And when this phenomenon of coalescence happens at the top of the foam, you observe also a decrease of the foam volume with time. So if you want to have a stable foam, almost stop these three main mechanisms of foam destabilization. But the main question is how to obtain stable foams. So you have different examples in the literature. You have the first one that is called like peak rim or Ramsden foams. And you can have stable foam because the solid particle will just adsorb at the air-water interface and form a solid layer surrounding the bubbles. And then you can really have stable foam during months or even during years with these solid particles. You can also use specific proteins when you want to design stable foams. For example, you can use the hydrophobin 2. So it's a specific protein because it's a very elastic layer at the interface, stabilizing the bubbles against coarsening and coalescence during months. You can also use some surfactants and more particularly self-assembly of surfactants, such as you can see here, vesicles, because these vesicles are completely crowded between the bubbles inside the foam liquid channel, so that slow down the drainage, the coarsening, and the coalescence. So then you can have very stable foam also with surfactant self-assembly. But the main problem with this very stable foam, it's like for specific application, you need to control the foam stability. So here you have a picture of a guy that is using a foam to decontaminate a surface. But the main problem is that at the beginning of the process, he needs a high quantity of foam, quite stable foam. But at the end of the process, he needs to destroy, to remove the foam. So he has two only possibilities. Either he removes the foam by hand, or he adds some chemical defoaming agent to destroy the foam at the end of the process. But the main question is, there is another way to control the foam stability. For example, if you have a surface to decontaminate, you put a foam on the surface, the foam acts on this surface, but then if you can use an external stimuli, for example, such as drawn here, a magnetic field to destroy the foam on demand, then the guy just needs to remove the small quantity of contaminated liquid remaining. And you don't need to use chemical defoaming agents, which can be very harmful from the environment, to destroy the foam at the end of your process. But the scientific challenge is how to produce this kind of responsive foams based on green surfactants to avoid the use of chemical defoaming agents. So our idea, our strategy was to use as green surfactants a specific fatty acid. It's the 12-hydroxystearic acid. So it's a fatty acid with 18 carbons and an hydroxyl group on the 12th position. And these fatty acids come from the castor oil plant. And what is very nice with castor oil, it's the only one vegetable oil that is a non-edible oil. That is to say that you don't have any competition between the food industry to feed people and the chemical industry in this case. And this fatty acid is already well known and used to gellify solvent because it forms self-assembled. The tube structure, we use small angle Newton scattering experiments. So in small angle Newton scattering experiment, the neutron beam is scattered by the tube solution, and you just measure the evolution of the intensity as a function of the wave vector q. So this wave vector q just depends on the scattering angle here and on the wavelengths. So here you can see the scattering curve obtained for the tube solution, and you can see on the scattering curve the presence of three peaks, three bright peaks. And this peak shows that lamellar phases are present in solutions. That is to say that the tubes are not empty tubes, but they are multilamellar tubes, as drawn here. So they are micron-sized tubes with multilamellar tubes. And so from the position of the peak, you can determine the interlayer spacing between two bilayer inside the tubes. And from the information on the large wave vector part, you can determine the bilayer thickness of your bilayer inside your tube. Then we follow the response of these tubes as a function of the temperature. And what we observe by optical microscopy, it's like tubes that appears as rod here in black rods. We observe tubes until 55 degrees, but exactly at 60 degrees for this system, we don't see any more tubes, and the solution is completely isotropic. So again, we use small angle Newton scattering experiment, and here you can see the scattering curve at 60 degrees in red And you can see that the curve is completely different from the previous one. You don't see anymore the bright peaks. And we can easily fit this curve with a form factor of a sphere, showing that spherical micelles with a diameter of 45 angstrom is present at 60 degrees. So these big micron-sized multilamellar tubes transit precisely at 60 degrees into spherical nanometrical micelles. And this transition between the two states is completely reversible. So we have thermoresponsive fatty acid assembly from this specific packing parameters, and that leads to this change of micron-sized tubes into spherical nanometrical micelles with the temperature. Then we wanted to see what is the structure of these tubes at the interface, because we want to design some responsive aqueous foam. So if you want to stabilize a foam, you need to have a foam stabilizer at the interface. So the main question was if these tubes are just unfolded at the interface and form a simple fatty acid monolayer, or if these tubes can go to the interface and absorb below this fatty acid monolayer and act like solid particles in the case of Pickering foams. So to answer this question, we use neutron reflectivity experiments. So in neutron reflectivity experiment, the neutron beam is reflected at the air-D2O interface, and you just measure the reflectivity coefficient as a function to the distance to the surface. So here you can see the reflectivity curve obtained for the interface between air and the tube solution. On this reflectivity curve, you can clearly see the presence of smooth interferences fringes. And from the position of the fringes, we determine that the thickness at the interface is around 300 angstrom. So this is markedly higher than just a simple fatty acid monolayer of around 21 angstrom. So this shows that tubes are not just unfolded at the interface to form a simple fatty acids monolayer. Then we try simple models to explain the results. So first, we wanted to see if these tubes are not just unfolded at the interface and form lamellar phases with the distance between the lamellar phases of around 300 angstrom. So from this lamellar phase model at the interface, we calculated the profile of the fatty acid fraction according to the distance to the surface. And from this profile, we calculated the corresponding reflectivity curve that you can see here in black. Then we compare this lamellar phases model with our experimental data in blue. And as you can see here, the two curves are very different. The interferences fringes are very too high in the case of lamellar phase at the interface. So that clearly shows that tubes are not unfolded at the interface to form simple lamellar phases. So then we decided to model the absorption of tubes at the interface. So we developed a mathematical model of tubes adsorbed at the interface below a fatty acid monolayer. And in this mathematical model, we took tubes with exactly the same dimensions that we determined previously in bulk by microscopy sense experiment. And the main parameter in our model is the interlayer spacing and the number of tubes adsorbed at the interface. From this model, we calculated the corresponding profile of the fatty acid fraction according to the distance to the surface. Again, from this profile, we calculated the corresponding reflectivity curve that you can see here in black. And we just compare, again, our experimental data here in blue with our model of tubes adsorption here in black. And as you can see here, the two curves are very similar, showing that tubes are adsorbed at the air-water interface and can stabilize the interface in the same way that solid particles act in the case of Pickering foams. Then we wanted to study what is happening with the temperature, because I showed you previously that tubes turn into micelles with the temperature, and we wanted to determine what is happening at the interface. So we just recorded the reflectivity curve at 60 degrees, as you can see here in red. And we don't see anymore the presence of interferences fringes, and we can clearly easily fit this curve with just a fatty acid monolayer at the interface. That is to say that when tubes are no more present in bulk, when they transit into spherical micelles, they are no more present at the interface. We just have a single fatty acid monolayer. But remarkably, by decreasing back the temperature, tubes are reformed in bulk and they are reabsorbed at the interface. So the transition between tubes into micelles is both reversibly at the interface and in bulk. So then we wanted to see what is the forming properties of these tube solutions and can adsorb at the air-water interface. So first we just produce a foam by hand shaking a tube solution like this very easy experiments. And as you can see here, we observe quite high quantity of foam with this tube solution. Then when we compare a picture of foam taken just after foam formation and after 6 months of conservation of the same foam at room temperature, we can see that the foam volume didn't change and the bubble size almost didn't change. So with this simple fatty acid system, we obtain ultra-stable foams. Then to understand how the foam is stabilized, we just stain a tube solution with a fluorescent probe, as you can see here in bulk, with confocal microscopy experiment. We produce again a foam by handshaking from this stained tube solution. We took few bubbles of the foam, and we observe again by confocal microscopy the presence of tubes inside the foam liquid channel in very high quantity. So tubes are present at the interface surrounding the bubbles and in very high quantity in the liquid phase. So it's really the presence of these tubes that stabilize the foam and leads to this ultra-stable character of the foam. Then we wanted to see what is happening at 60 degrees because the tubes turn into micelles at 60 degrees. So we just produce the foam, put the foam in an oven at 60 degrees. And as you can see, we observe very fast foam destabilization. In a few minutes, we don't have any more foams. So the spherical micelles can't stabilize the foam. So then we decided to see what is the evolution of the foam stability during temperature cycles. So for this, we just produce foam by bubbling gas into a tube solution until a fixed volume of 45 milliliters is reached. And when the temperature is at 20 degrees, tubes are present inside the foam. The foam volume remains constant with time, as you can see here. But by increasing the temperature to 60 degrees, tubes transit into spherical micelles inside the foam, and we observe very fast foam destabilization. But remarkably, by decreasing back the temperature to 20 degrees, tubes are reformed inside the foam, and we observe that the foam volume remains constant again with time. So with this simple system of fatty acids, we obtained the first temperature tunable foams 10 years ago. So the main problem with this system was for real industrial application was like the temperature transition between tubes into micelle was at 60 degrees, and it was still too high for a lot of industrial application. So then we decided to study how to tune and to decrease this temperature transition to lower temperature. So the first parameters that we studied was the effect of the nature of the counterion. So for this, we just decreased the chain length of the counterion from 6 to 2 carbons. Again, we determine the temperature transition between tubes into micelles as a function of the carbon number of the counterion. And as you can see here, by decreasing the chain length of the counterion, we increase slightly the temperature transition from 60 to 70 degrees. So yeah, when you change the nature of the counterion, you can slightly tune the temperature transition, but it's still too high for a lot of industrial application. So then we studied another parameter, the effect of the molar ratio, and we defined the molar ratio as the number of mole of the fatty acid divided by the total number of mole inside our system. And here you can see the result for the counterion with 2 carbons. So here on the graph where the temperature is plotted as a function of the molar ratio, you can see that at low molar ratio where we are in excess of counterion, the temperature transition is around 42 degrees. Then you observe a sharp increase by increasing the molar ratio until a plateau is reached around 78 degrees. So we observe 3 regimes as a function of the molar ratio for the tubes to micelles transition. And we can tune the temperature transition by tuning the molar ratio. Then we did exactly the same experiment for all the counterion from 2 to 6 carbons. And as you can see here, we observe again the same trend with the 3 regimes, but the transition is shifted to a lower temperature by increasing the chain length of the counterion. For example, for the counterion with 2 carbons, at low molar ratio, the temperature transition is around 42 degrees, and for the counterion with 6 carbon is around 20 degrees. So you can now, just by tuning the molar ratio and the nature of the counterion, you can really have a wide range of temperature transition between tubes into micelles. But then we wanted to determine what is happening at the molecular scales that can explain this change at the supramolecular scale for this temperature transition between tubes into micelles. So for this, we combine different type of experiments, different cell scanning calorimetry experiments, wax experiment, infrared spectroscopy measurement. And the conclusion is like at low molar ratio, you have an excess of counterion. So the pH is markedly above the pK of your fatty acid. So all the fatty acids are under ionized state. At low temperature, the alkyl chains are under gel state. So you observe the formation of tubes. But when you increase the temperature, the alkyl chain just transit from the gel state to the fluid state. And so you have a change of packing parameter to lower packing parameter and higher curvatures. That is why you obtain the formation of these nanometrical size micelles. Then when you increase the molar ratio, at high molar ratio, you have an excess of fatty acids in comparison to the quantity of counterion. So the pH is close to the pKa of the fatty acids. At low temperature, so you have a mixture of fatty acid under ionized state and protonated state that interact by hydrogen bondings between the head groups. And you have the alkyl chain in a gel state. You increase the temperature, you have the alkyl chains that transit from the gel state to the fluid state, but you have still the head groups that are bound together due to the hydrogen bonding. So you have still tubes in solution. But then at very high temperature, the hydrogen bond just can just be broken. So you have a surface melting that change the packing parameter, and you have the formation at high temperature of spherical micelles. And here the second regime is just an intermediate regime, because when you increase the molar ratio, you increase progressively the quantity of protonated molecule inside your systems and the quantity of hydrogen bonds inside your systems. So it's really the change at the molecular scale from the alkyl chain state, from the gel state to the fluid state, or at the head group level that can explain the different molecular interaction and this different self-assembly morphology and the wide temperature range for the tubes to micelles transition. So in summary, just by tuning the nature of the counterion, you can tune the temperature transition from 60 to 70 degrees. But the best parameter is really the molar ratio, because by tuning the molar ratio and the nature of the counterion, you can have a wide range of temperature transition from 20 to 75 degrees. And then we check that we can really have temperature tunable foam for this wide range of temperature. And yeah, we can have temperature tunable foam from 20 to 75 degrees. You just need to choose and to combine the right counterion with the right molar ratio, and you can destabilize the foam on demand at the temperatures that you want. And this is very more useful for a lot of industrial applications. Then the next scientific challenge was how to produce photoresponsive foams based on green surfactants without chemical modification. Why we were interested to develop photoresponsive foams? It's because light as an external stimulus has many advantages in comparison to temperature, because with light you can direct light at a precise location with a micrometer scale resolution, and you don't need to touch the sample in comparison to temperature. So our strategy was to use a chemical engineering approach and to use the thermoresponsive fatty acid tubes that I just showed you previously with specific colloidal particles, such as carbon black particles. Why these particles? Because they are known to act as heat generators. That is to say that they can absorb UV light, and due to a photothermal conversion effect, they can transform the light into heat. And so for this system, we use tubes with a temperature transition around 45 degrees. So then we produce some foams, and we had a look at what is happening under UV irradiation. So here you can see the control foam just with the tubes. So this is the picture before UV, and then it's the picture after UV radiation from the top of the foam after 7 minutes. And as you can see here, nothing happened. The foam is still stable upon UV radiation. But when we do exactly the same experiment with the foam based on tubes and the mixture with carbon black particles, you can see that just after 20 seconds of UV radiation from the top, you observe very fast foam destabilization. And after 1 minute of UV radiation, you don't see anymore the foam. So the presence of these carbon black particles really destabilize the foam upon UV radiation. But what is happening? So to understand what is really happening, we use an infrared camera to record the evolution of the temperature inside the foam upon UV radiation. So here on this picture, you can see that the temperature of the initial foam was around 25 degrees. And for the foam with the tubes and the carbon black particles, we can see that after 20 seconds of open UV irradiation from the top, the temperature inside the liquid foam channel increased around 45-50 degrees. That is to say, above the temperature transition from tubes to micelles from the system that we choose for this study. And for the control foam just with tubes, even after 6 minutes of UV radiation, the temperature of the foam remains almost constant around 25 degrees. So really upon UV radiation, we can clearly see that the temperature inside the foam liquid channel increases due to the presence of these carbon black particles. So if I summarize the mechanism of foam destruction by UV, so before UV, between the bubbles, you have this high quantity of tubes and carbon black particles. You apply UV radiation from the top of your foam. The carbon black particle just absorb the UV light and transform this light energy into heat. And so the temperature inside the foam liquid channel just increase above the temperature transition between tubes and micelles. So you don't have any more tubes to stabilize the foam. You have just spherical micelles. So you observe very fast foam destabilization. Then we decided to use, instead of these carbon black particles, to use magnetic particles. And why is because it's known also that magnetic particles are intense absorber of light irradiation. And as you can see here, with magnetic particles, we can also destabilize easily the foam upon UV radiation. But what is nice by using magnetic particles, it's like you can also destabilize the foam on demand by the application of a magnetic field. As you can see here, we just applied a magnet on the side of the foam, and we can destabilize easily the foam. So by combining this kind of magnetic particles with the fatty acids tube, we obtain the first thermophotoresponsive foams. And another approach that we used to design this kind of photoresponsive foams was based on the fact that the fatty acid assemblies are known to respond to pH changes. And so we just combine these fatty acid assemblies with photoresponsive components, which are known to induce pH changes under UV radiation. As photoresponsive component, we use a photoacid generator, the diphenyliodonium nitrate, that is known to get photolyzed by UV light to form an acidic moiety. So you apply UV light and you observe a pH decrease. So for this study, we just start with a solution of fatty acid at a high pH, pH 12. So we have spherical micelles inside the solution. As you can see here, we have a limpid solution. And by signs, we can clearly see that before UV radiation, we have very nice spherical micelles. Then we just put this sample under UV radiation, and we observe a decrease of the pH from 12 to 9, and a transition at the macroscopic scale from a limpid solution to a turbid solution with a slight yellow color. And then by TM, what we can see is that we have the presence of micron-sized tubes, and by small angle Newton scattering, we can clearly see that on the scattering curve, we can see the bright peaks that I show you in the first part of my talk. So we recover these big micron-sized multilamellar tubes. So it's really the UV radiations that trigger the self-assembly transition from the spherical micelles, in this case, to the multilamellar tubes. But we wanted to understand what is happening at the molecular scale that can lead to this transition at the supramolecular scale upon UV radiation. For this, so we use infrared spectroscopy measurements. And here you can see that before UV radiation, we have just one peak corresponding to the presence of fatty acid under ionized state. After UV radiation, you can see two peaks. So you have still the first peak corresponding to the fatty acid under ionized state, but you have the second peak corresponding to the presence of protonated fatty acid molecules. So before UV radiation, all the fatty acid molecules are under ionized state. You just put your sample upon UV radiation, and you have a mixture of protonated, deprotonated fatty acid molecules. So upon UV radiation, you have the photoacid generator molecules that release some protons that interact with the deprotonated molecules to form some carboxylic acid. So if we summarize the link between the molecular scale and the microscopic scale, so before UV radiation, the pH is markedly above the pKa of your fatty acid. All the fatty acids are under ionized state, and they lead to spherical micelles and a limpid solution. Then you put your sample under UV radiation, the photoacid generator releases some protons, interact with some ionized molecules, and you observe a decrease of the pH, now close to the pKa of your fatty acid, and you have the two states of fatty acid, protonated, deprotonated ones, and interact by hydrogen bonding. And so you have the formation of your big micron-sized multilamellar tubes, and you observe a tubed solution at the microscopic scale. So then we determine the foaming properties of this system. So before UV radiation, we have spherical micelles. You observe very fast foam destabilization. The spherical micelle can't stabilize the foam. But after UV radiation, tubes are formed inside the foam, and we observe very stable foam again. So in this case, we obtain pH photoresponsive foams, so we can stabilize foams that were previously unstable just by the application of UV light. So in summary, for the first part of my talk, just by using a specific fatty acid, the 12-hydroxysteric acid coming from the castor oil plant, you can have thermoresponsive and pH-responsive fatty acid assemblies. And by combining them with specific particle or specific components, you can design multi-responsive foams. That is to say, foams that you can destabilize on demand by temperature, UV, or magnetic field. And the potential application of these multiple responsive foams include on-demand defoaming in industrial processes or washing and material recovery processes. So now I want to show you how, by using the other strategy of colloidal assembly, the directed assembly approach, based on fatty acid soaps, you can design ultra-flexible magnetic filaments. So first, how to make assembly of nanoparticles. So again, you need colloidal assembly. So you have the self-assembly approach. So the nanoparticles can self-organize in 1 dimension, 2 dimension, or 3 dimension structure. Or you can use the directed assembly approach. So you use an external field to drive the particle assembly into well-defined shape. You can use magnetic field, electric field, or light. And from this particles assembly, then you can design some stretchable conductors, soft chain microacquator, and so on. But the main prerequisite when you want to design self-reconfigurable material chains and networks based on nanoparticles is like you need to bind permanently together the particles. Otherwise, when you just remove the applied field, such as, for example, magnetic field, the particle will just fall apart. And then it's impossible then to design from this directed assembly some self-reconfigurable material chains and networks. But the question is how to bind permanently together nanoparticles. So nanoparticles can be bound permanently together into stiff chains and not flexible chains through electrostatic or van der Waals attraction. As you can see here, it's nanoparticles under an electric field. You can obtain flexible chains, but you need to use micron-scale particles, so no nanoparticles, and through soft molecular linkers, such as surface-grafted DNA or polymer, as you can see on these 2 examples. So you need synthetic chemistry method. So the main problem is that you have flexible chain, but based on micron-scale particles. In our case, we want to use nanoparticles. So the scientific challenges was how to bind permanently together nanoparticles to then obtain flexible chains and soft reconfigurable materials without synthetic chemistry method. So the question was to find some soft linkers based on green materials, which can be flexible linkers if you want them to design some soft reconfigurable materials, and linkers that can be also responsive to external stimuli if you want to have some smart materials. So our idea, our strategy to try to find this kind of soft, flexible, and responsive linkers was to have a look at reconfigurability in nature. And the best known example of reconfigurable material in nature is sandcastle. So sandcastle is made of sand particle. You just put a little bit of water. The water weighs the surface of your particle. When you just put into contact the particles, you have the formation of capillary bridges that hold the particles all together into complex shape. And then when you just dry, or the sun just dry your particle assembly, you recover some sand particles. So our strategy was to use this kind of capillary forces at the nanoscale in bulk to bind permanently the nanoparticle. Liquid will weight the surface of the particles, and just by shearing, when the particle will just enter into contact, they will just form capillary bridges and form some capillary suspension. So here you can see on this picture an example of micron-sized particles dispersed in a solvent. And when they just add a little bit of water, the water was immiscible with the continuous phase of the solvent. You have the formation of the capillary bridges between the particles, and you can see the drastic change of the rheological property from the fluid-like to a jet-like state. So our idea was to use these capillary forces in bulk to bind permanently the nanoparticles. But until our study, there was no proof that the capillary forces can work at nanoscale. It was always done just with micron-sized particles. So the first objective was to determine how to form capillary bridges between nanoparticles in bulk. From a theoretical point of view, it was possible, but from an experimental point of view, there was no proof until our study. And then was how to obtain flexible chains and soft reconfigurable materials based on these capillary bridges at the nanoscale. So the idea was to have some nanoparticles, to apply an external stimuli, to obtain flexible filaments and network due to the formation of these capillary bridges between nanoparticles. So our strategy to achieve this aim was to use iron oxide nanoparticle, manganese nanoparticle with a diameter around 20-30 nanometer. Why magnetic particles? Because we wanted to use an external magnetic field to drive the particles assembly. And then it was just to disperse this nanoparticle inside the fatty acid soaps aqueous solution based on fatty acid assembly. Why again fatty acid soaps? Because they are green surfactants. They have some affinity to iron oxide surface. They are responsive to temperature, as I showed you in the previous part of my talk, due to the transition between the alkyl chain in gel state and the fluid state. And they are solvosurfactants, that is to say, when the fatty acids are under their carboxylic form, above their melting temperatures, they behave like oil. So they can be the second immiscible liquid inside the water to weight the surface of the particles and form the capillary bridges. And when you just use the fatty acid in the ionized state, you have the surfactant properties of the fatty acid. And so if you use, as I showed you previously, fatty acid assemblies with fatty acid under carboxylic form and carboxylate form, you can veiculate the carboxylic form to form the capillary bridges. So when you just mix the nanoparticles with fatty acid soap, what we observe is the formation of fatty acid capped nanoparticle with a lipid shell surrounding the particles. So then we use a directed magnetic assembly field to produce our filaments. So for this, we use a multi-directional magnetic field. As you can see our setup, so it's 4 electromagnets. And in the middle, you have the sample chamber. We put the dispersion inside the sample chamber, and we just follow the particle assembly by optical microscopy. Here you can see a picture of the nanoparticle and the microaggregate inside the fatty acid soaps self-assembly dispersion. We just turn on the magnetic field in this direction, and you can see that the magnetic field just pull all together the nanoparticles and microaggregates along the direction of the applied magnetic field, and they form some filament structure. Then we just turn off the magnetic field. So we remove the field-induced attraction and we relax the extended chain. And what you observe when we turn off the magnetic field, it's the filament structure persists after switching off the magnetic field. So there is something that binds permanently the nanoparticle together. And you can clearly see that the filaments are very flexible because they are undulating inside the surrounding medium after switching off the magnetic field. So to understand how does it work, how the filament structure persists after switching off the magnetic field, we did a simple experiment first. So we took a microscopic glass slide. We put a hydrophobic spacer in the middle, as you can see here. And on one side, we put nanoparticle capped with fatty acid. And in the other side, we just put nanoparticles in water. We apply the magnetic field in this direction, like this. We observe the formation of filaments for fatty acid capped nanoparticles, and nothing happened for nanoparticles in water. We turn off the magnetic field. The filament structure persists for nanoparticles with fatty acid, and nothing happened for nanoparticles in water. So this simple experiment just shows that it's really the presence of these fatty acids surrounding the nanoparticles that bind together the nanoparticles permanently, and that's why you have still the filaments after switching off the magnetic field. So then to understand the structure of these filaments, we use different types of microscopy experiments. So first we use atomic force microscopy experiments. And as you can see here, we can see that the filaments are very thick because the thickness is around 50 to 100 times thicker than just one single nanoparticle filaments. And the particles are embedded in quite thick shell of fatty acids. Then we use TM experiments. So we stain the fatty acid solution before with uranyl acetate. And now the fatty acid appears in white, as you can see here, and the nanoparticle in black. And you can clearly see that each particle is surrounded by a thick shell of fatty acids. Then we have a closer look by cryo-TM, and you can clearly see that between two nanoparticles, you have the formation of capillary bridges between the nanoparticles. And you can see that the fatty acid shell seems to be quite fluid surrounding the particles. So to better understand the effect of this fluid layer surrounding the particles, we decided to study the effect of the fatty acid phase, the alkyl chain, when the alkyl chain are in gel phase or in fluid phase, on the nanoparticles assembly. So for this, we use differential scanning calorimetry experiments. We determine the temperature transition between the gel phase and the fluid phase as a function of the carbon number of the fatty acid soaps that we use to produce this kind of filaments. Then we just put nanoparticles for all the fatty acid in a gel phase. We apply the magnetic field in this direction, but we didn't see the formation of filaments. So that shows that when fatty acids are under a gel phase, they don't lead to the permanent assembly of the nanoparticles. But when you perform the same experiment for all the fatty acid soap in a fluid phase, you always obtain the formation of these filaments when you apply the magnetic field. So this clearly shows that the fluidity of the absorbed fatty acid layer surrounding the particle is really the prerequisite for the permanent chain formation, because if you don't have fluid layer, they can't form the capillary bridges that bind all the particles together. But we use the fact that you have this transition between gel phase and the fluid phase to control the disintegration and the formation of these filaments. So here you have an example of filaments that we produce at 45 degrees when the alkyl chain were in fluid state. We decrease the temperature below the phase transition. So you have now the alkyl chain in a gel state, and what you can see is like the temperature acts as an external stimulus because it just collapses the structure. You disintegrate your filament just by tuning the temperature. And then if you increase again the temperature to 45 degrees and you apply again the magnetic field, you can recover your flexible filaments. So it's really this crystallization inside your nanocapillary junction that leads to these filaments disintegration. So these nanocapillary bridges are responsive to temperature. Then we studied the property of these filaments in a one-dimension time switch field. So here you can see that we produce first filaments along the direction of the applied magnetic field. And in the second picture, I just changed the direction of the applied magnetic field like this. And you can see that automatically all the filaments just bend and try to follow the change of field polarization. And you can see that they all bend and form at the end self-closed structure. So all the linear filaments turn into well-defined structure, and this self-closed structure remains even after switching off the magnetic field. So how we can explain that the linear filament just curl when you change the direction of the applied magnetic field in a one-dimension time switch field? It's quite simple. So you have the filaments with the AN and the BN, and you have two electromagnets. And the filaments has only two possibilities, the trans or the cis curling pathway. So here you have the north that is going here, the south that is going here, and the same here. So the AN and the BN can go just to the opposite direction to follow the change of field direction, or they can go to the same direction like here. And when the two ends just meet at the middle of the filaments, when they just touch each other, they just stick to each other due to the capillary bridges and the self-linking properties, and you obtain the formation of infinity structure. But when the two ends just meet each other like this, they just form kind of rings or square structure. So, and this is really due to the self-linking by the nanocapillary bridges. And so we observe at the end some rings or some square, depending on the bending modulus of our filaments, some infinity structure, or sometimes some heart or even hook. And the structure really depends on the mode, the frequency, intensity, and duration of the applied magnetic field. And then to study also the flexibility of this magnetic chain, we just put one single chain in a two-dimension magnetic field. So like this. And as you can see here on this picture, we can see the formation of one curl and even two curls, and the chain doesn't break. So that shows the very high ultra flexibility of this chain. And why? It's because all nanoparticles are surrounded by a fluid layer of fatty acids, so they enable a perfect rollover of the particles from each other. So you have very, very high flexibility of this kind of filaments. So if I summarize, we just use iron oxide nanoparticles, we disperse them in fatty acid aqueous soap solution, and you obtain fatty acid capped nanoparticle. Then we just put these fatty acid capped nanoparticles under a magnetic field like this. And so the magnetic field just pull all together the nanoparticles are microaggregate. When they just touch each other, they enter into contact, they stick to each other due to the formation of capillary bridges. But what is nice with these capillary bridges based on fatty acid, they are ultra flexible soft connector that leads to perfect rollover of the particles, and they are responsive to temperature. So you can form and reform these capillary bridges on demand by tuning the temperature. So now I just want to show you some example of soft materials that we can design from these filaments and their assembly. So here you can see some picture from a micrograbber. So this micrograbber is just based on the accumulation of flexible filaments. And we just control the motion of this micrograbber by the intensity and the gradient of the external magnetic field. And as you can see here, when we put this micrograbber with some flexible chains surrounding him, by just moving the micrograbber, when it just touches some filaments, like you can see here with his hand, the filament just stick to his hand due to the formation of, again, capillary bridges and the stickiness of these capillary bridges. So just by moving this micrograbber, he can just grab all the filaments surrounding him. So it's just an example of what kind of microrobot you can design from these sticky and magnetic filaments. So another responsive soft materials that we can design from these filaments is ultra soft magnetic network. So to design this kind of network, we just added another electromagnet at the bottom of our setup, as you can see here. We put on the top of this magnet so the dispersion of iron oxide nanoparticles and fatty acid soap. We just turn on the magnetic field in this direction. You obtain the formation of upright standing chain. You turn off the magnetic field. Due to their weight, the chain just fall down. And when they just touch each other, they just stick to each other and they form a very nice percolated soft network, as you can see here. But the property of this network, it's a self-repairing network. That is to say, you have your initial network, you just cut your network, for example, as you can see here on this picture with a sharp stylus, and you have a rupture network. But then by turning on the magnetic field, all the filaments can just, touch each other again. And when they touch each other again, they just stick to each other again, and you have a regenerated network. And you can turn off the magnetic field and you recover your initial network. And this is really due to the fluidity again of the capillary bridges and their stickiness. The filament can be really broken and reform on demand. So I hope that I convince you that, yeah, you can really use capillary forces at the nanoscale to bind, for example, nanoparticles. And by using fatty acid soap, you can really have very soft and sticky capillary bridges that leads to very ultra flexible filaments and ultra soft networks. And these capillary bridges based on fatty acids are thermoresponsive, so you can control the disintegration, the formation of this kind of filaments. And the potential application of all this system is as micromanipulator and soft robotics. I show you two examples with the self-repairing network and the micrograbber. So it's the end of my talk, and I really hope that now you are convinced that fatty acid soap are just more than soap to wash your hands. And you can really, by using colloidal assembly, so by using self-assembly approach or directed assembly approach, you can really design multi-responsive soft materials. So I would like to thank all the people who participated to this work. So in France, in the US, in Germany, in Argentina, and all the funding that I received from my institute, the French National Institute of Agricultural Research on the Environment, and from the European Union. And for sure, I want to thank you for your attention. And again, I would like to thank the AOCS community for this award. I'm very, very happy to receive the award, and merci beaucoup. Now it's time for questions.
Great. Well, thank you very much, Anne-Laure, for a fascinating talk and actually very easy to follow. And it's just the possibilities just keep on coming to the head. So I found myself like, oh, I better write down some questions because I was listening too much. So thank you for that insight. That was really amazing. I thought fatty acids were old school, but I think there's a lot we don't know about it. We have a couple of questions, and I have, of course, questions. So let me read the first question here in our Google Sheet. Have you tried to make the foams at 60 degrees Celsius where you initially would have a different interface stabilization instead of heating the foams afterwards? I guess that was in the transition between the spherical micelle and the tubes.
Yeah. So yeah, we did exactly the same. We produce also the foam directly at 60 degrees with the micelles, and it's very unstable foam. So, but what I didn't tell you during my presentations, it's due to the transition of self-assembly from tubes to micelle, but also because you have a change of this fatty acid monolayer surrounding the bubbles. Because with the spherical micelles, you have the alkyl chain of the fatty acid under a fluid phase that don't stabilize so well the interface. And in the case of tubes, you have the fatty acid with the alkyl chain in gel state that form very solid fatty acid monolayer at the interface. So I simplify a little bit for my talk, but yeah, it's not just only due to the presence of spherical micelles, but also of the properties of this fatty acid monolayer at the interface. But the most important parameter is really this transition between big micron-sized tubes that completely gellify the foam liquid channel, and then you have so small spherical micelles that lead to this drastic change of stability.
No, thank you. I see a question just came in right now. I'm assuming it's related to this part of the work. And I was also interested in the fact that you did it with hydroxysteric acid, which is a wild and wonderful molecule. But can you do any of this with other fatty acids? Did you try oleic steric acid, or do you require the special structure of hydroxysteric acid?
So we try with other fatty acids, but they all form other kind of structure like vesicles or worm-like micelles and so on. And so you don't have this kind of gel inside the foam liquid channel, because with 12-hydroxysteric acid, you have these big micron-sized tubes that then, due to these big tubes, they really form easily at low concentration a gel inside the foam. So if you try to put some water on the top of your foam, the water can't enter because you have really a gel inside the foam. And in the case of vesicles and so on, you need to use very, very high quantity to try to gellify the foam liquid channel. And so you don't have the same destabilizations and process. So it's really due to this specific fatty acid that leads to this specific self-assembly in water, like it does also in oil. It's really like, yeah, fascinating molecules. So yeah.
So sorry, and I'm going to jump in here. So if you would put that hydroxyl group in maybe another position, the 12-hydroxy, it's probably going to be different, right?
I know that it works for the 10 hydroxysteric acid, but we never try for other kind of hydroxylated fatty acid. But yeah, maybe, yeah.
Oh no, because these days some people are turning oils into through they epoxidize it, and then they turn into polyols, and then they make polyurethanes. But one would be able to convert vegetable oils into polyols with different positions quite readily and cheaply. I was just wondering if so if it does with 12-hydroxy, maybe it's not as specific as oleogelation, because if I remember well, 10-hydroxysteric acid does not gel. So
in our case, it's because the 10-hydroxysteric acid form also micron-sized tubes. That's why it works. And with almost the same dimension, so big micron-sized tubes in length.
Okay, so here comes a question that follows this discussion. Is there another fatty acid other than 12-hydroxysteric acid with similar property of tube formations?
You have some bola lipids and some other lipids, but what is very nice with 12-hydroxysteric acid, it's cheap, it's already used in a wide range of application. And so I don't see the point to use other specific fatty acids that you need to do with synthetic chemistry method with just only 1 milligram and so on. So what is nice with this one is like you can really apply this directly for industrial application. So yeah.
Right, and just the audience, remember that you make most greases with 12-hydroxysteric acid and stuff. So and it's from castor oil, so it's widely available. I mean, just whoever's hydrogenating it, that's where you get it from. But it would be interesting to know if there's anything else that does it. Okay, another question now in part two of the talk. I had a question about the structure. You showed very interestingly how pH was so important and the counter ion was so important and how you could tune the pH via all of this. What is the pK of? Did you ever measure the pH?
No, we don't know exactly the pK of this fatty acid. So this is always the main problem with fatty acid in water. So, but we know that it was close to the pK of this fatty acid, around 9.5. So that's it. But we don't know precisely the pK of the fatty acid. It's always this big question about fatty acid soap. So
yeah, that's why I was asking. I just, you can see something reported, but I don't think anybody really knows what pH is. But what about the variation in pH? Did you know from where to where you went? Or you?
Yeah, always it was between 9 and 12, because below 9 you begin to observe some crystal and no more fatty acid tubes.
And how important was for that part of the work the cation, the counter ion that you added in there? Is that really critical? Like, it's really critical what you put it in, right?
Yeah, because it completely controls this temperature transition between tubes into micelles. So I should just show you some model counter ion. But for example, the counter ion with 2 carbon, it's called ethanolamine. And this one is widely used in a lot of industrial application, for example, in cosmetic application for hair coloration products. But we can also have these tubes with amino acid, for example, and it works very well. But the same, it changed the temperature transition between tubes into micelles. But yeah, this is why just with kind of model counter ion, but you can have really green counter ion, because this one were not so green, but you can just use arginine, lysine, choline, and so on, and it works very well also.
That would be very nice to see some choline, for example. That would be fantastic, or an amino acid. That's great. That's great.
Yeah. So yeah, we did this and we published this also.
Oh, wonderful. Now, on part two, as a question submitted, are there any organic molecules that can improve capillary bridging between these fatty acid-derived particles? Are there any organic molecules that can improve the capillary bridging?
I don't know why we need to improve these capillary bridges, because they are already very nice, I think. But yeah, you can use, I think, other kind of molecules. But what is not so easy to form capillary bridges at the nanoscale, it's like you need to form very small droplet of your immiscible liquid. So it's like kind of oil in our case, because we work in water, and it's not so easy to have very small droplet, because you have already some nanoparticles. So you need to have oil droplet that is smaller than your nanoparticles. That's why with this fatty acid it was very nice, because we are just using the carboxylic form of the fatty acid that are at the molecular scale, so they are smaller than the nanoparticles. With micron-sized particle, it's easier, because you have big micron-sized particles. You can just emulsify quite well your second immiscible liquid oil, and then you can wet the surface of your particles. But with nanoparticles, to have something smaller than the nanoparticles, oil droplet, it's very difficult. So
understood. And well, maybe the organic solvent could help turn a fluid into a smaller particle.
Yeah. But then what we wanted, it was really to have something that can lead to soft reconfigurable materials, flexible and responsive linkers. And then with solvents, you can't have this kind, I think, of responsive linkers and so on. And green linkers also. We were trying to do some simple work always based on green molecules. So yeah.
Maybe I missed it, but the fatty acid droplet that you put in would be like a little micelle, I'm assuming, and that would hit the particle, and then how does it spread with the carboxyl side onto the particle, or the chains onto the particle?
Yeah. So we assume, we suppose that the first layer is completely anchored at the surface of the iron oxide particles due to the chelatin and coordinating affinity with the iron oxide magnetic particles. And then you have this first layer that completely wets the surface of the particle and change the physical chemical properties, and then can induce then the condensation of the other molecules.
So it's a multi-layer.
It's our hypothesis.
Did you ever scatter X-rays over and prove that there's multi-layers of these fatty acids or no?
We tried with neutron scattering experiment, and it was really not so easy, because you have still a lot of fatty acid soap remaining in water, and so even by centrifugation and so on, it was really not so easy. So
And sorry, and the last question for me, this is capillary bridging. So it's the fatty acid monster covering the particle, and then there's water in between the particles, right? Is the water responsible for capillary bridging, or it's the fluid fatty acid self-assembled touching another fluid fatty acid self-assembled?
No, it's really the two fluid fatty acid shells that when they touch each other, they form these capillary bridges. So you have like fatty acid capillary bridges, all bridges, and then you have water in the two sides of these capillary bridges.
But there's no water involved
inside. No.
Not water capillary.
No.
No, thank you very much. And let me just see if there's any other questions. No, that's all the questions we have for now. So that was a great discussion. I mean,
yeah, thanks a lot for all the very nice questions. Yeah.